Hello. Oh, welcome everyone.
Hi, thanks everyone for coming, and thank you for being patient. I just wanted to make a small joke before we began, and to let you know, Kirsten and I, my colleague, we come from Pacific Northwest National Laboratory. P&L cybersecurity really works. We can explain that joke later.
Okay, welcome everyone. We're so glad to have you here for our session on the future potential of metabolomics for illuminating the molecular dark matter of soil. Thank you all for coming. This is a really important and emerging area. We have an incredible panel here of speakers lined up for you. And in the end, we're hoping to have a conversation about what the field could look like. This is really a very new and emerging area. So we're looking forward to the diverse perspectives that'll be presented today and hearing your perspective during our conversation. Yes. So the metabolome is critical to planetary carbon cycle. The atmosphere has about a third of the carbon as the soil. So 3 times as much carbon is stored in the soil as the atmosphere. Much of that carbon has been lost by human activities through erosion, through respiration. As you can see in this slide, carbon leaves the atmosphere through photosynthesis and enters into the soil as carbon. It can also be removed back to the atmosphere through decomposition or soil respiration and plant respiration. So one of the major goals in soil ecology and climate science is to push this pump of photosynthesis to increase carbon going into the soil and then find mechanisms by which we can retain that carbon in the soil rather than having it be respired back into the atmosphere. Metabolomics plays a major role in this, as you can see in the image on the left, in that as that photosynthate is coming in the roots, the question is how will roots and microbes and soil interact in order to create stable carbon that can then be sequestered over the long term. And this is in planetary science, it's been called the carbon dioxide removal, and the idea is to remove carbon dioxide from the atmosphere through photosynthesis, pump it into the soil, and figure out ways to retain it in the soil over the long term. So this is one piece of the climate mitigation solution. We need to figure out how the metabolic interactions between plants, microbes, and the stabilization on the soil can bring this to bear, and how much carbon we can actually store over very short time periods. And Joëlle will talk more about these rhizosphere or root interactions. Part of the challenge is that soil is one of the most complex materials on the planet, and this is because of all of the complexity. There's biological complexity in that there's millions of microbes that live in that soil. There's chemical complexity in the soil organic matter. You can see here one soil profile. There are many different kinds of soils that have different mineral and organic interactions. We've got physical structure to the soil that form in aggregates and pores. This changes the hydrology, and it's variable with the different kinds of mineralogy. We've got hydrologic, spatial, and temporal variability. So this makes it difficult to come up with generalizable solutions. From a chemistry perspective, one of the grand challenges is how do we remove metabolites from the soil in a way where we don't have extraction bias or other biases and actually capture the active metabolites and differentiate that from metabolites that might be stabilized in the soil. And you can see here there's lots of ways that plants and microbes can interact with the soil in order to increase carbon sequestration. Much of that depends on microbial metabolism and plant metabolism, and that's a major frontier. Typically what we do, this is a image from one of Myrna's recent publications, and she'll be talking more about ways that we might be able to approach this challenge. Typically we have an environmental stressor or process that we're trying to isolate the metabolites from. We do metabolic measurements, as you can see in the workflow, the identification, data analysis, pathway analysis, and then ecological interpretation. And while this conceptual model looks very elegant, there are many steps along the way where we have challenges, and Myrna will talk more about that. This is just a quick example from research that's coming out of our lab group where we can see that most of the metabolite features detected in soil are unannotated. So this is a network analysis of the metabolites that we have pulled out of a rhizosphere, so the root area, the soil that's adhering to the root. And what you can see here in the black And the black dots are all the metabolites that are detected but unannotated. So we don't know what any of that is. And many of them don't even cluster structurally in a way where we could start to think about what class of molecules they are. The green dots are the ones that are annotated, and we have an understanding of what their function is in the soil. So there's a huge opportunity here for us to move forward into the dark matter. And Sean will talk more about potential approaches for trying to understand what's going on in these complex systems. And with that, I'll pass it to Tom.
Thank you. So as Kirsten was mentioning, when you perform a metabolomics analysis of soil, you really return a lot of unannotated features. Everyone that does metabolomics knows that this is probably the current grand challenge in metabolomics. How do we annotate structurally everything that we detect? So if we do clinical type assays of blood and cells, we have some proportion of data that's unannotated. It's even worse in soil because of the richness of soil. There's a lot of polymeric substances. There are also a lot of abiotic processes that occur due to interaction with metabolites with mineral content. And so if you're all familiar with metabolomics analysis, this is just a cartoon showing an example with mass spectrometry. We introduce the samples into the instrument, we'll perform a measurement, we have a lot of peaks, maybe it's accurate mass alone, ideally it's accurate mass in conjunction with tandem mass spectrometry. We take all of that information and we match it to what we have in our existing reference libraries. And the biggest challenge with that paradigm is we only can identify what we've seen before. And what that means is we take usually compounds in pure form that we buy from chemical suppliers, we run them through our instrument, whether it's mass spec or NMR. We take those profiles, those spectra, those tandem mass spectra, other properties that might be measured, and we put that up into the reference library, and then we perform an analysis of a complex sample, and we match the data to the reference library. And where there's a match, we say, yes, that's glucose, or this is alanine. The challenge with that is it's really hard to get authentic, pure substances for every molecule that we might encounter. We already know it's challenging on the clinical side, but in soil, again, even more challenging just because of the breadth of chemical diversity that might be present there. If I were conducting a metabolomics analysis and I encountered an unknown peak and I wanted to identify it, I would not use the typical high throughput paradigms of LCMS or even proton NMR profiling. Ideally, what I would do is I would enrich the substance and use more conventional, more traditional approaches to structurally identify it, whether it would be conventional structural elucidation using NMR or X-ray crystallography or microelectron diffraction. That's all well and good, but that's a slow process. It's not high throughput. You also usually require, significantly more quantities of the substance than what you might require for a typical profiling, or you might need to grow a crystal, or at least kind of partially grow a crystal in the context of micro-ED. So something that we would like to propose to this group, and at the end of the presentations we will have a group discussion, is should we be more seriously considering a new paradigm, which would be reference-free metabolomics, or metabolomics where we computationally predict the properties of molecules as they behave in instruments rather than try and purify or purchase pure forms of substances to run through. If we could do that, I think that would really open up the field and really open up the possibilities, and it would be sort of like sequencing but for metabolomics in terms of the information content that we could obtain from our typical profiling studies. And Adam will talk a little bit more about some new technology that might play a role in that paradigm. So here's our agenda. This is the first presentation, the introduction and motivation for the workshop. As Kirsten mentioned, I think we've got a really exciting and diverse lineup here. You'll hear about different aspects to the challenge of performing metabolomics of soil, and also, again, the importance and the opportunity that is there in terms of being able to potentially force more sequestration of carbon into the soil to help deal with the climate effects that we're experiencing right now. And I don't know if anyone can smell smoke in the air right now, but I know I can from the wildfires that are currently burning in the north of here. These were the workshop objectives and learning outcomes that we listed for the workshop. You all ideally saw those when you signed up. I won't read through those, but I'll put them here. But I will point out that we would like to write a position paper on this topic, and everyone in this room is invited to become a co-author and contribute as much as they are interested in and are able to. And then lastly, I won't dwell on this. We can pop this back up again at the end of the presentations, but these would be some initial questions that we would offer everyone, both ourselves and yourselves, for consideration to get the discussion going. So with that, I would like to introduce our next speaker, Joëlle Sasse from University Zürich. I don't remember the title of your talk, but you'll see it soon.
Me neither. Thank you. Okay, hello everyone. While we are waiting for the slides, I'm very excited to give the first input talk of the first workshop of this exciting conference, and I hope you guys are more or less awake and ready to discuss with us afterwards. So I'm a plant biologist by training, and I kind of started in Switzerland working on plant transporters and then moved to the States to Lawrence Berkeley lab, actually in a microbial lab, and I didn't know anything about microbes really, and it was like a perfect combination of like interdisciplinary researchers to learn more about a general system. And we talk about one aspect of this, about carbon fluxes and how to increase carbon sequestration into soil. So what I would like to take you through now in the next couple of minutes is kind of the plant perspective on that. Obviously, if we talk about CO2 fixation, algae are also really important, or the marine plants, so to say, the marine systems. And they are highly relevant as well, but my expertise is more in plants, so we will be talking about the land aspect of that. I brought you some numbers here on this slide to just have some idea on where the carbon goes that is fixed. So the CO2 fixed, this would be around 100%, and then about 2/3 stay in the above ground part of the plant. About 1/3 is used for biomass growth of the shoot, and about 1/3 is respired again, so directly released again into the atmosphere. The remaining 1/3 goes below ground, and of that 1/3, or like overall 10% are respired again, 15% are used for biomass growth, and 5% of the fixed carbon initially is ending up in root exudates. Those are compounds that are synthesized within the roots and then transported outside. So they are in the rhizosphere and they are available for example, microbes to use as signals or as nutrients. So obviously, these numbers on how much carbon is, respired and how much is going to the shoot and root, this varies greatly between plants and also between ecosystems. So that's kind of an average what you're seeing here. The general idea is to increase the carbon input and sequestration and to decrease the carbon release, right? That's the overall idea. And if you just quickly think about decreasing the carbon release, we actually don't really have many options to do that. If you think about natural systems like grasslands and forests, we can only try to preserve or regenerate these systems so that they don't lose too much CO2. And obviously, in the face of climate change, where several ecosystems are thawing now, this starts to be an issue because then a lot of carbon is released in these systems. In agricultural systems, we can think about management practices, and it was shown, for example, with low tillage or mulch farming, or also by maybe using different types of crops, the carbon respiration can be lowered. So that's definitely something that should be taken into account and implemented, because agriculture causes about 10% of the greenhouse gas emissions globally. So that's a major factor that we should consider when thinking about that. But our workshop is more focusing on how to increase the soil carbon input. And I made a schematic drawing here on how we picture that conceptually. We have the plants that fix the CO2, right? And then we would like to increase the root to shoot ratio so that more carbon is transported below ground. Then we know that it's important on the type of root systems that we have. So the deeper the roots go, the easier it is to fix the carbon afterwards. So we need to think about the root system. Then the carbon is released as exudates, what's labeled here as labile carbon, because it can be respired easily. And then the carbon is taken up by microbes and finally stabilized on mineral surfaces, for example. So that's kind of the general idea on how we picture that workflow. The issue, though, is that this is not an easy problem to solve because there are many, many factors that play into that, and I have a slide on that later on. The issue is that each single one of these factors is not well researched, and we don't really know how they play with each other. So often when people try to increase the carbon sequestration, what they end up doing is increase the carbon respiration, so exactly the opposite. So clearly we understand the system not well enough that we can do that in a targeted and efficient way. So what are the factors that affect soil organic carbon formation and stability? This is by no means exhaustive, and it's also not a core competence of mine, so it's very well possible that your preferred factor is not on there. I would just like to mention a few of them just to give you an idea. So in general, soil organic carbon is affected by the type of climate we are in, the type of ecosystem we are in. Then from the plant perspective, on an ecosystem level, it depends how many different plant species we have and how they are distributed, how evenly they're distributed, and how much functional diversity we have among these plant species. It depends what type of root system they have, what root morphology, and how deep the roots go. Then regarding exudation, the rhizal deposition or the amount of metabolites that are produced and exported by plants are crucial in quality, so in composition, but also in quantity, like how much carbon is exported by different types of plants. Also, there are other types of cells released by roots. And there are other types of not soluble compounds that are released that are more polymeric, and also, for example, mucilage is an example like that. The carbon to nitrogen ratio is critical, we have found, in exudation in regard to how it's used by microbes and how stable it is in soil. Generally, when we talk about soil organic carbon, there are two ways. People talk about particulate SOC or soil organic carbon that is produced more from roots and litter and that is considered more unstable compared to the mineral SOC that is produced by microbes and considered to be more stable or easier to sequester and to fix in the soil. Then from the soil point of view, there are a couple of examples that we know are important. For example, the ratio of rhizosphere to bulk soil. So rhizosphere is the soil influenced by roots, right? The soil water holding capacity is important, the soil type more generally, and the balance between nitrogen and carbon, for example, is crucial. If you think about microbes, their diversity overall, their activity is important, and it was already mentioned in the introduction that the mycorrhiza, which are symbiotic fungi, are critical, and there are different types, the arbuscular and the ectomycorrhiza, and each of them have a different activity regarding how much carbon they turn over and how much they are involved in stabilizing carbon. Then obviously also the type of minerals that we have in soil are important, and overall this influences how much carbon is fixed and how much is lost. And you might have known about the priming effect, where people try to sequester carbon, but then respiration is actually induced mostly by microbes, and a lot of carbon ends up going to the atmosphere again. So those are some of the factors. Overall, I think this was mostly to illustrate that we are looking at a very complex issue, right? And it's very multidisciplinary. We touch on plant biology, microbiology, soil science, geology. We probably need some kind of modeling approaches to put all these factors in context, and also we have to think about these dynamics in the face of a changing climate. So, it's interdisciplinary. We have a complex structure of different factors that we need to integrate somehow. We need to think about natural systems such as grasslands and forests, but also about agriculture. And then when we think about like scientific approaches to try to understand that issue better, we need to figure out a balance between how much can we use like reductionistic approaches in the lab and how much do we have to look at natural systems and how much can we extrapolate between the two. My core expertise is in exudate studies, so I just wanted to give you a hint of what we try to do there and to kind of illustrate that this is an important point to look at. So, you know, exudation is the process on how carbon gets into the soil from the roots, and we understand very little about its dynamics. We only recently published a paper with some of these, with some of the new findings in there, and one of them is the idea that plants probably, or they probably produce a set of compounds that is well made by all the plants that we call the core metabolome. And the idea is that this core set of compounds are produced by all the plants, by all the roots to interact with the core microbiome that is associated with plant roots. So those are probably compounds that are there always. And then there are the compounds that are more specialized, that are probably more important more for specialized plant-microbe interactions. And that should be looked at specifically in a context, for example, of a forest in comparison to a grassland. Also, exudation is very distinct depending on the environment. If you look at the middle plot, that's a principal component analysis plot that was shifted a little bit, unfortunately. But you can see like the green dots on the right-hand side, and those are plants that were grown in the presence of clay, and they very clearly have a distinct exudation profile. What happens is that clay binds a lot of the compounds that are exported by plants, and we then found as well that the microbes can release some of these compounds again that were bound to clay. So clearly we have this very complex interplay with minerals as well that has to be taken into account, and also plants obviously react to the presence of different types of microbes or also different physiochemical properties of soils. So plants are responsive to their environment as well. One key aspect for us in the lab to try to understand exudation is to look at the more molecular level and to investigate transporters that are involved in the in exudation of these different compounds. And why this is important for us is that we gain a method on how to regulate exudation of specific compounds. Because by up or down regulating these different transporters, we can look at increased or decreased flow of certain metabolites and then understand how this impacts, for example, interaction with different types of microbes. Or then translated to carbon sequestration, maybe we can upregulate specific transporters to increase carbon sequestration by a specific factor. So I would like to end with a couple of open questions. And to start with the central maps that are displayed better on your screen, I'm happy about that. So what you can see is that the red areas are high, for example, in soil carbon in the upper map, or in respiration in the lower map. And what you can see is that the areas that are high in soil carbon usually are low in respiration. So there's a negative correlation between respiration and soil carbon, which is not very surprising. But what you can see as well is that we have a lot of local dynamics going on. So the question is if we can even think about these problems as a kind of a global problem, or if you have to look specifically in different locations. If we start with the questions now, and we start like from the plant perspective and exudation, then one of the major questions we ask ourselves in the lab at the moment is if we study exudation in a sterile hydroponic setup, which we often do because we have a low metabolite background, we have only plant compounds without, for example, microbial compounds being there, how much can we extrapolate from the fingerprints we see in this hydroponic setup to a soil environment, right? And this is both true for the composition of the exudate, but also for the quantity. Then if we look at the root system, we have a lot of spatial and temporal dynamics. Not all the roots do the same over their development, so that's another factor to consider. Then plants react to biotic and abiotic stresses around them and adjust the profile and as well as the amount of carbon they produce. So that's a crucial factor to take into account. And then we already touched upon the issue that we need to think about exudates and microbiomes and minerals in the soils as well, and that probably in a local context. Also, there are plant-plant interactions. They react on the presence of their neighbors. So that's, for example, crucial in grasslands, right, where we have a lot of the same species. And then from a more like conceptual point of view, I touched on that already, is how can we reduce this complex system that we see now to a point that we can actually learn about the input of the different factors and then extrapolate again from that to the big system we have. So how do we choose good model plant species? How do we choose good synthetic communities, for example, where we limit the amount of microbes to a few that we can handle in the lab? Do we use fabricated ecosystems for that, for example? How do we explain the priming effect? People did some studies on that where they found that it depends if you want to sequester carbon and you just kind of pour it onto the soil. It matters if you apply that once or continuously over a certain amount of time, and also it depends if you apply one compound to the soil or a diverse mixture of different compounds. Then how much, like thinking about, you know, the global issue of climate, of a changing climate is, we need to like realize, or we need to understand how much carbon we actually need to sequester to have an impact on a global level, right? Like how much CO2 do we have to remove from the atmosphere to see an effect on our global temperature, for example, and which ecosystem is actually able to do that, right? Like how do we do that realistically? What do we choose? What scale do we need to address that if you want to have an impact? And also maybe as a more applied question is, can we maybe construct that in a way that we can use the sequestered carbon as a renewable energy source, right? For example, as a biofuel. So I would like to leave you with that, and I'm looking forward to our discussion later on.
Yep, thank you, Joëlle. So that was a really interesting look at all of the factors that are at play in the entire ecosystem that might contribute to how carbon flows into the soil and how it might stay there. Now we're going to hear from Myrna Simpson from University of Toronto, Scarborough, who's going to tell us how hard it is to actually understand what carbon is actually in the soil.
All right, thank you for the introduction and thanks for organizing this workshop. I'm really pleased to be here. So I'm going to focus more on the chemistry of soil and some of the potential challenges related to using metabolomics to better leverage mechanistic information, especially when we're trying to elucidate some of these complex soil processes. So as we've heard already, a lot of the soil carbon is very dynamic and participates in a number of cycles. And all of this carbon is in the form of soil organic matter. And soil organic matter is not only important for the local soil biogeochemical cycle of not just carbon, but nitrogen, phosphorus, and a whole bunch of other elements. It's also a huge part of the global carbon cycle. And soil organic matter is necessary for maintaining soil health. So it's a source of nutrients. And as we start to lose soil organic matter, we start to lose soil fertility. So it is a huge issue. Loss of fertility is going to impact our ability to maintain crop yields, et cetera, et cetera. Soil organic matter also has a lot of physical benefits to the soil. But from a chemistry perspective, it's also the most chemically active portion of the soil per unit mass. Now, soil organic matter also participates in a lot of pollutant interactions. So this is both like heavy metals, organic pollutants, and it's also important for the transport and sequestration of those pollutants. So soil organic matter, again, is really important for a number of reasons. Because of that, soil organic matter has been studied for a really long time, so over 300 years. You can actually go back into the literature and read all about it, and there has been a really interesting evolution of knowledge in terms of what is the chemistry, how reactive is this material. Now, more recently, we've undergone a complete paradigm shift in terms of what soil organic matter is actually made of. And that's highlighted in this figure from a review in Nature from 2011. In the top panel, you can see that historically, because of the lack of high-resolution methods, researchers concluded that soil organic matter was made of humic substances. And this was an accepted theory for decades and decades. In the late 1990s, with improved techniques such as NMR, high-resolution mass spec, et cetera, researchers were actively probing the chemistry of soil organic matter using these advanced techniques. None of these techniques could actually find any evidence for humic substances. So this is the paradigm shift that I'm talking about. The modern understanding of soil organic matter is that it's a complex mixture made up of identifiable biomolecules. So if we have a look at that complex chemistry, as shown here, we see that there is a molecular continuum of soil organic matter. This continuum includes small molecules all the way up to really large biopolymers, including proteins and a whole bunch of plant-synthesized biopolymers. So when we talk about a complex mixture, we're not only talking about acids and aldehydes and things like that. We're talking about differences in polarity, differences in molecular weight, a whole bunch of things. Now, from a metabolomic perspective, what's also important to point out is that there are a number of compounds that can be assigned specifically to plants and some assigned to microbes, but then as shown in the middle of that diagram, there's a whole bunch of things that come from multiple sources. So this is important from a metabolomics perspective if we're trying to trace processes, right? There are some molecules that can come from a whole bunch of different sources, and they may not be informative. Also, from thinking about, if we're thinking about metabolomic strategies, we have to always start with the extraction and the methods we want to use. So this slide just highlights different functionalities that would be important to consider for any type of extraction. And I know this is a really big table, but essentially what it's highlighting is that different components within soil organic matter have different polarities. So you have really hydrophobic things, and you have more polar compounds that can be extracted in water or buffer. You have compounds that are pH sensitive. You have compounds that have different charges. They can be positively charged or negatively charged depending on the soil pH. So this chemodiversity is really challenging when we're trying to develop strategies for extraction and isolation. And then, of course, there isn't one analytical method that can manage all of these. If you have really nonpolar compounds, then LC is not a good technique for that. You have to automatically use GC and things like that. So there's a lot of considerations that need to go into working through a workflow for metabolomics. Now, in addition to that, a lot of these compounds will undergo various abiotic and biotic reactions, so they can be transformed. As shown on the side of that diagram, a lot of these compounds can also bind to the inorganic portion of the soil, the clay mineral portion. So I'm just going to briefly talk about these, just to, again, give you a bit of a sense of how transformation and absorption processes can also impact what we're trying to accomplish with a metabolomics approach. So here's some example diterpenoids. These occur in conifers, and their transformation is very well documented. So they can undergo both abiotic and biotic transformation. So again, depending on the experimental design or the hypothesis, you may need to factor in, again, isolating all of these different molecules just for that one kind of portion. In addition to that, components can be preserved and not degrade at all. So you're often looking at a combination of untransformed compounds in addition to their transformation products. And again, that's if you're trying to capture the entire class. Now, transformation can also happen to the more biopolymer style compounds, or I guess biopolymers in general. Here's an example with lignin. Lignin side chains can also be altered through biotic and abiotic processes. So again, if you're trying to isolate a biopolymer, you're going to have all these side chains that are going to have different functionalities, such as changes from acids to aldehydes and things like that, as shown here. Now, when we think about mineral interactions, again, this is a really detailed slide, but it just highlights that the clay minerals are really important for the attenuation of a lot of compounds. And in this slide, it's shown that there are a number of processes that occur between organic matter compounds and the clay minerals. And this can often render them non-extractable. There's also evidence that soil organic matter can interact with itself. So you want to think of more of a layering type phenomena, but again, that is dependent on the type of soil, the type of organic matter, and also the mineralogy of the soil. But to give you a sense of how this interaction may limit extractability, here's some data of specific plant-derived compounds. The dark bars are just the yield of extraction, and then the white bars are after we remove the minerals with hydrofluoric acid. So you can see the yield of those organic matter compounds was significantly increased when we got rid of those minerals. So we know that this interaction with clay minerals renders a lot of these compounds as non-extractable. And also in my lab, many years ago, we carried out a sequential extraction. I think we used 4 or 5 different extractions. And we could only resolve between 5 and 10% of the soil organic matter. So again, that gives you a sense of how strong these interactions are. So the last topic related to soil organic matter chemistry that I just want to touch upon is the time related to how fast some of these processes occur. So we have these dynamic processes that are not well understood, and this is something that metabolomics can really provide unique and insightful knowledge about. So you want to think about soil as a hotbed of biology and chemistry that's going on simultaneously, but we don't really have a good understanding of some of the metabolites that cycle fast and some of their processes. If we have a look at this slide, again, it shows that there's not a lot happening, not a lot being represented in terms of rapid cycling processes. So this is something that we have an opportunity to address and could potentially shed some light on this. But you can see that there are varying timescales and residence times for different classes of molecules. So we know that there are some chemistry and biology constraints that are controlling these processes. So lastly, just some challenges from my own perspective. I think we need to come together as a community and develop robust and reproducible extractions, because I think that's going to be important because soil itself is really heterogeneous, and we have to make sure that we have some kind of standardized approaches. I think defining metabolite sources is also important. so that we can make sure that we're defining mechanisms that are related either to abiotic or biotic processes. We need to have a consensus with respect to the analytical approaches and standardization of instruments. And then, of course, how does all this wonderful information provide us any insight on ecosystem function, especially with the perspective of carbon? So essentially, we will likely have to come up with integrative approaches because it's going to be difficult to come up with one method that's going to give us all the information that we need. So with that, I would like to thank you for listening, and I will take questions during the panel discussion.
Thank you, Myrna. Next is Sean Brady from Rockefeller University, and Myrna just talked about the challenges in getting carbon out of the soil. Now, how do we know what carbon should be in the soil? What should we expect? And so Sean's research really digs into some of these aspects and tools of what could be in there, what to look for.
All right, thank you very much. A disclaimer from the very beginning, I don't do metabolomics. So you can either fall asleep during my talk, or you can decide you're going to hear about metabolomics for the next 3 days, and this is an interlude about something else for a little while. So what are we interested in? We're interested in the biosynthetic dark matter. Is there a hidden reservoir of small molecules like this hidden in the soil microbiome? And so I think that the thing you have to understand to get my talk is just this one fact, that all of those molecules on that previous slide come from biosynthetic gene clusters. They come from unique groups of genes that contain all of the genetic information needed to make each of those molecules. Very easy to find in a sequenced bacterium, not so easy to figure out what's out there in the soil environment. So that's what we think about, and that's what I'm going to talk about. The traditional approach for trying to understand what's going on in the soil is, of course, you culture bacteria. You put them in fermentation flasks, you use some type of metabolomics, or use NMR, whatever technique you're going to need to characterize the metabolites are there. There's two drawbacks. One is we don't culture most of what's out there, so we can't characterize their molecules using that strategy. The second drawback is even if you culture them, you don't turn on most of the biosynthetic genes that are present in those microbes. So we've missed most of what's out there. We take culture-independent approaches where we extract DNA directly from environmental samples, we clone that DNA, we sequence it, we look for new gene clusters, and then we have a couple of different platforms for trying to turn that into molecules. You either dump it right into a cultured model system to see if you can use synthetic biology to turn that gene cluster on and access the molecule that's encoded by it, or more recently, and maybe analogous to the idea of untargeted mass spec, we predict the structures that are coming out of that, and we synthesize them by total chemical synthesis as a way of getting at molecules you've just never seen before. I'm not going to spend the rest of my time talking about that because each one of those stories still today is relatively one-off. You've got to figure out what to do with each gene cluster. What I'm going to spend the rest of my time is just talking about how do we see things through sequencing? How do we see what's out there? And the dream is shown here, that we would just go in, we would extract DNA from the 10,000 different bacteria that are in a soil sample, we would sequence it, and we would have all of the gene clusters that are there. That is computationally impossible still today. If it was possible, it's too expensive, and so no one's done it on any type of scale. What we've relied on instead are these types of strategies, simple one-gene marker gene analyses, where you identify a single gene that's common to some biosynthetic family. You look at that gene in the environment and you use it as a marker for what types of biosynthesis might be out there. Very cheap to do, computationally easy, and scalable. If you know how species analysis is done for soil samples, that is, use 16S analysis as a marker for what types of bacteria are there. We do the same thing as a marker for what types of chemistry is out there. And the reason it works is that as much as we think bacteria are smart, they're not that smart. Right? They know how to make molecules in about a half a dozen or maybe a dozen different ways. Then they use those same genes over and over and over again to make most of the chemical diversity that's out there. Some really rare things, that's true, but most of what we're going to find, at least coming from bacteria, is part of one of the half dozen or dozen major ways that bacteria have learned how to make molecules. On that slide, you see lots of different molecules, but they come from two main families. They come from polyketides, and they come from non-ribosomal peptides. These are all made from acetates. These are all made from polymerizing amino acid or amino acid-like structures. They're a family because all of the gene clusters that make those molecules use the same types of genes. So all of the polyketide biosynthetic systems use basically the same domains. They use them in different ways. They use them with different substrates. They use them with different numbers. It means that what we can do is we can design degenerate primers that recognize those conserved domains. We can amplify them out of the environment. You don't just get one gene, but you get thousands of genes. Next generation sequencing allows you to sequence that very easily. All of a sudden you have millions of tags that are markers for what's going on out in the environment. And the bioinformatics for that is very simple. We develop a phylogenetic tree of all the sequences we see here. We map onto them all the known ones. And if you want to see whether your soil sample makes a rapamycin-like molecule, well, you look for a rapamycin-like marker gene. And if you want to know if it makes a rifamycin-like molecule, you look for a rifamycin-like marker. If you want to know what makes something new, you look for soils that are rich in markers you haven't seen from cultured bacteria. We and others have written a variety of programs that allow you to do that on large scale. And this is just some of the data you can get out of experiments like that. This is, the left-hand side is a little bit of pop science, I'll argue. I mean, it's us looking at 300 parks in New York City and mapping the biosynthetic diversity that's there, what's hidden in the urban park samples. And the take-home from that is we could find markers for every different type of natural product that's made it to the clinic that was originally discovered by scanning the globe for cultured soil microbes. We can find in individual soil samples in parks in New York City. Just says the huge diversity of microbes are there and what PCR allows us to see deeply into the biosynthesis that's there. This is maybe a little bit more intelligent. It's mapping diversity across a large landmass. So it's mapping biosynthetic diversity across all of Australia. And we were looking for what drives differences in soil microbe biosynthesis. And the only correlation we can find is latitude. So things at the same latitude, no matter where they are across Australia, largely make the same types of molecules. We've seen the same thing across the United States, and maybe it's not surprising because that's how large animals, that's how plants, that's how the rest of the globe is set up, and so it's probably how biosynthesis is set up. So we believe that's probably going to hold true as you look at soils around the globe. What do we really do? We use that information to guide our discovery of molecules. We find soil samples that have interesting markers, we clone that DNA, we find gene clusters that are interesting, and we use one of our two platforms to characterize a growing number of metabolites that are out there that have interesting activities. Again, the ultimate goal, and I'll get there in a few slides as to where the field is to get us to here, where we can just sequence everything, and then it's just a matter of characterizing the molecules. There's an intermediate step, and that is, can we do cheap things like PCR that give us more information about what's going on out there? Can we learn more than one domain in a gene cluster? Can we learn multiple domains in an individual gene clusters out in the environment very cheaply and on scale so we can learn a little bit more about what's hidden in the soil sample? And that's what this is representative of. So each one of these is an individual domain into a network. That's one gene cluster, but we can do it cheaply. We don't have to do it by full sequencing. And that the idea really comes out of what happens when you try and understand what's going on in the soil microbiome. You extract DNA from dirt, you clone that DNA, and to saturate what's going on in a dirt sample, you need about 20 million clones. So you need 20 million, 40,000 base pair clones to begin to saturate the biosynthetic diversity that's out there. Ideally, you'd like to array that as one little clone in each well of a 20 million arrayed library. We can't do that, but we can array it at about 20,000 clones in a well, and therefore you can condense your library down to a manageable unit. And then we sequence for these marker genes in each well. And what you realize is there's more information. As soon as you array a library, there's a bunch of information you can pull from it. And so what you find is that if two genes were part of the same gene cluster out in the environment, they appear at a very high frequency in the same wells in the library because they just happen to get captured together. So if two markers appear together, it means they appeared together out in the environment. If they don't appear together, it means they didn't appear together out in the environment. So just the co-occurrence frequency of these biosynthetic genes in a library allows you to begin to predict what actually exists out in the world connected to each other. All right, so you can develop the networks, and we can see hundreds of them deep into soils. It's called concat-seq. You can begin to compare that to what we know. So you compare those networks to all of the things that have been characterized in the past, and the only things that look like known things are those little red guys at the top. Everything else is unknown. All of this is unknown biosynthetic diversity that you haven't seen before. Why is that? Well, we can go back in the library and just hand pick the clone that has each of these networks and just sequence that clone. It works out, it actually validates the networks. We really are looking at gene clusters, and they really are, when you see the whole gene cluster, unrelated to things that have been seen before. You can make predictions of where this diversity lies, and it lies deep in the metagenome. Most of the biosynthetic diversity is really, really rare out there, so none of the shotgun sequencing approaches have been able to see it. It would take about 2,000 human genome equivalents of sequencing a single soil sample to begin to see the biosynthetic diversity in the deep metagenome. Doesn't mean you can't do it, but no one's going to spend that money at this point to sequence a single soil sample to see what's really out there. And this approach allows us to do that, do it on scale. We can compare soil samples. So this is just looking at 4 different soils, and now looking at their deep biosynthetic metagenome. You can compare the networks from one soil sample to the next. And again, this is just a weird way of showing it, but nonetheless, most of them are blue, a little bit of red in each soil. That's all that's known. All of the blue stuff is unknown. What's shared between any 2 soils, that's the gray bar. There's very little shared between 2 soils. What's shared between 3 soils, almost nothing. What's shared between 4 soils, essentially nothing. All right, so not only is it deep, but it's very different from sample to sample to sample. Again, we don't know how many soil samples you'd have to go through before you start seeing redundancies, but at least on a small scale, most of what's out there is different from environment to environment. We were interested in just the common stuff. You'd expect the common stuff to be known. So you pull out the cluster, you express it in the lab, get this molecule, which is called omnipeptin. It's a novel molecule. It has 2 really rare building blocks that have never been seen before. So even the common things in biosynthesis are things we haven't seen before. All right, I'm going to finish up with one last little thing, which is this is the dream. I've told you kind of stepwise over the past decade or so of what we've been doing and other groups have been doing to try and deeply see what's in the metagenome. We would really like to get here, right, where you can see everything, and the only problem is how do you express these things? Well, I would say basically Q4 last year, the first quarter of this year, we finally have the tools to at least imagine that dream. It's not because anyone cares about the soil metagenome. Fortunately, they don't really, right? It's because people, right, there's a small number of us, probably all in this room, who really care about the soil metagenome. It's because people care about getting to a $100 human genome. And of course, huge advances in sequencing. And the generation 3 or 3rd generation sequencing, where you can read not 100 base pairs, then have to stack those together to try and assemble 10,000 different bacteria out of a metagenome, which has been impossible. But instead, these machines can read tens of thousands of base pairs in long stretches, where now you can see complete gene clusters where you don't have to assemble them. It really revolutionizes how we're going to see metagenomes. Still expensive. Costs about $200,000 probably to sequence a single soil metagenome, but that data is going to be real, and it's going to tell us what's out there. And so we and others have, of course, really jumped on that in the past 6 months or so, and a huge amount of metagenomic sequencing data is coming out. It will be revolutionary in the next 5 years. There's no question we will have a much better picture of what's going on in the soil from a biosynthetic perspective. My last 2 slides are to say it does present another problem, which is probably a good problem, which is now that we can see everything potentially, if you have a 1 million member library, how do you go back and find that clone that you want to go look at? Seems like a stupid problem, but it's one of the big bottlenecks in the field of how do you go back and find the thing you want. So we just developed a new cloning strategy, which we've called KISS cloning, that allows you to do that. So if you're ever making a metagenomic library, you should make it with a KISS cloning vector. And so it's basically a barcoded vector that has a toxin gene on it. You can use CRISPR to introduce into that library a single guide that will only survive the clone you want. Happy to describe that in more detail, but it basically allows us to sequence the library, and then with a single transformation, just allow the single clone you want to come out of there. It means we can now look at hundreds of biosynthetic gene clusters out of these libraries. We're scaling, we'll see what happens in time, but I really think this is the future, some version of that where you're directly sequencing the metagenome and rapidly recovering things out of it, which we can now see. I'll stop there. This just comes from a review article that just came out from us, and it basically is here to tell you that I think the first part of this is kind of the dream of what you'd like to be able to do. The first part of it, the left-hand side, we probably know how to do pretty well as a field now. This part, generating molecules from these things, will be next over the next 5 to 10 years, and that's, I think, where metabolomics will come in. We come in, how do you detect these things? How do you look for them? How do you characterize them on a large scale once you actually have the gene clusters that are present out in the environment? And that's where I'll end, and happy to answer questions in the session later.
Awesome, thank you, Sean. It's a little bit of a sobering presentation on some of the remaining challenges and just the volume of data that can be generated through those approaches. For our last presenter, I'd like to introduce my colleague, Dr. Adam Hollerbach from Pacific Northwest National Laboratory, who will tell us about some new technology that might be able to help interrogate the chemical diversity and universe in soil.
Okay, well, thank you very much. I will also preface my talk by saying that I'm not a metabolomics person either. The closest I really get to soil is maybe rolling around in it when I'm building instruments. But I'm still excited to tell you about some new technology that I've been working on with my colleagues at PNNL and trying to help try and understand and characterize what are some of these unknown compounds in soil that are giving people so many issues. And so this talk is about a SLIM Orbitrap. SLIM is an acronym for a type of ion mobility spectrometer, and it's an ultra high resolution one at that. And hopefully that we can use this data from the SLIM and also from high resolution mass spec to help characterize these unknowns, maybe without needing standards. And so just a bit of a background, I've heard quite a bit that people like to use LCMS or GCMS. They're very powerful tools. And there's even an ion mobility session going on next door. And so what I'd like to talk to you today about is not necessarily a replacement, though perhaps it could be one day, but really I'd like you to consider ion mobility maybe as a complement to a regime that you may be working with and see, hey, maybe if you run your analyses and there's something you can identify, maybe this could help you. And so just a little bit of background for ion mobility. Ion mobility is a gas phase separation technique. It looks at ions and it separates them based on how big they are. It's their size and also the electrical charge on them. And so this is a very common example that people show of an ion mobility spectrometer, probably the most common one, which is a drift tube, which is comprised of a set of stacked rings. And what happens is that you apply an electrical potential or a voltage gradient to these rings, and ions will follow that potential. They start from high and they go to low voltage, and then your ions will collide with the background gas and they will separate based on how big they are. So the small ions can kind of squeak through the background gas more easily than the big ones, which kind of crash into everything on their way through. And then they separate based on what we call a collision cross-section, or more scientifically, it's a rotationally averaged ion neutral collisional cross-section, which is shown at the, I put a little diagram at the bottom there, just showing kind of what happens as your ion increases in size. And you can see this interaction area increases in size as well. And this is one of the properties that can help differentiate certain compounds that maybe something like an LC may not be able to give you all the separation of, or the same thing with a GC. Maybe you can't separate everything. Maybe ion mobility can help you do some of your analyses. And so what in particular can ion mobility do that might be of benefit? Well, it can separate isomers. So it could be either positional isomers, if you have a methyl group on some of your metabolites and it's on the left-hand side, maybe there's a ring over there, or maybe there's a straight chain on the right-hand side, and then the methyl group is dangling off that side. Ultra-high resolutions, or plainly speaking, just high resolutions, can help you differentiate those. And so what I'm showing here are just a couple of really good examples that show isomeric differentiation with ion mobility. On the left-hand side, there's some sugars. Here, these are sodiated species that you may end up seeing in your mass spectrum. And they all weigh 365.1, or at least that's their monoisotopic mass with sodiation. And as you can see with this ion mobility spectrum, this was actually acquired with one of our platforms at Pacific Northwest National Lab. You can see that there are not only baseline separation between lots of these isomers, but there are also different isomers within each sugar that you can see. So there's some sort of either a rotation or maybe something is axial or lateral inside one of those chair conformations. And so we can see those, and then that helps try to help you identify all the compounds. Of course, then there's the question that, well, if one peak turned into two, does each one of those peaks that makes up the two also transform into other peaks? So that's something we think about and try to look at. The other thing that I wanted to show were some lipids. This is something that I have been working on quite actively for the last little bit. And these are just showing some drift tube measurements. So can you see a difference between the isomers that some lipids can do? So for example, if you have two different chains in a phosphatidylcholine, and is one of them an 18 carbon chain and the other one is a 16 carbon, and does that switch positions inside of the phosphatidylcholine structure? And you can kind of see a little bit of separation with those, at least with the drift tube, although I believe these are extracted from, these spectra are extracted from individual runs. They're not a mixture, which is probably fairly important for the complex mixture of soil. You can't just be like, oh, I want to look at this one and then throw everything else out, although maybe there's a good extraction procedure out there. That'd be pretty awesome. But I'm not aware of one. So you can at least try to think of, okay, if we can separate these with a really high resolution, can we look at that in a complex mixture as well and help you characterize to fulfill what you're looking for in your structure or in your soil sample? You can look at things like, boy, this is really hard to see. You can also look at cis-trans conformation. So do you have a cis fatty acid chain? So that will include a kink in your fatty acid structure. Well, if you rotationally average that, you try to spin it around, at least in three-dimensional space, that gives, you know, a slightly smaller structure than maybe a trans conformation would. So the trans tend to come out later. And so that gives some amount of separation that you can see. And again, there's a little bit better separation of what you can see by switching the chains in a PC in this particular example. But again, this just shows that this type of identification is possible, especially if you can pull those apart a little bit better. And so these are just some examples that are shown using, on the left-hand side is actually a high-resolution separation. On the right-hand side is a lower resolution. But what I'd like to talk to you guys about is a relatively new technique. It's been around for about 10 years now, but it's starting to become mature, is a technology developed at Pacific Northwest National Lab, whose acronym is SLIM, but that stands for Structures for Lossless Ion Manipulations. Really what it is a set of electrodes that you pattern onto some printed circuit boards, and you apply voltages to these in such a way that you can move ions and do things with them. Like you can focus them, you can send them around long paths to do ion mobility separations, you can trap things, you can do a lot of different things with them, hence the structures part. The lossless ion manipulations is that there tends not to be very many losses with these. Lossless doesn't mean zero losses. It means that we don't detect really noticeable or at least significant losses over time. And so the way that SLIM work is that they have, again, these pattern electrodes onto printed circuit boards, and instead of applying a voltage gradient, which if anybody's ever tried to apply high voltages across long distances, you can't really do that for any substantial length of an instrument. You end up approaching the breakdown potential of whatever it is that you're working in, be it at 1 torr or atmospheric pressure, and you end up getting a lightning storm, which is not good. And so what we end up doing is we apply these AC waveforms to some of these segmented electrodes we have on our SLIM devices, and these end up forming traveling waves, kind of like ocean waves, if you want to think about it that way. They can have high amplitudes, they can have low amplitudes, they can move really fast or really slow. And what we do is ions will move with the motion of these waves, and the smaller ions, for example, will be pushed a lot further with one wave than a big, large ion, which may move very little per one cycle, and therefore it'll take many cycles to come out. And there are several advantages to doing it this way. One is that you don't have a voltage limit that you have to work around. You can apply the same amplitude traveling wave, and you can go as far as you want, or as long as a distance, and you can still get an ion mobility separation. That's primarily what we use SLIM for. I won't bore you to death with the details of all the technical things about SLIM, but I would just like to say that you can send ions around very long paths. And so in traditional drift tube ion mobility spectrometers, or maybe some other ion mobility spectrometers that you've heard of, typically you might think of maybe a 1 meter separation. And that's, if you stretch your arms out, that's probably your wingspan. That's how big the tube is. What I'm showing you here is a picture of one of our SLIM boards. We have 2 of them, but this is a picture of, this is the bottom one, I think. And in total, if you can see that ions will travel this path of these gold-plated electrodes. And they'll go left to right, up cross over and then go down, so they can go around turns and go over this snake-like path. And if you were to take this path and stretch it out, this turns out to be approximately 13 and a half meters of a separation distance that you can send ions across. And really the board is probably no bigger than this podium. It's probably a little bit smaller. And so you can really get these long separation paths in a really compact space. And what you can also do is that you can end up taking the ions that arrive at the end of this printed circuit board and send them back to the beginning again. And then you can go through another 13 meters of separation. And if you want, if you're still not resolving your isomers, you send them back again and again and again. And you can end up doing really, really long separations that help you probe some of these intricate details that maybe some other techniques may struggle to do. And so what I'm showing here is that, how far can we actually kind of take these separations to hopefully see some unique features that maybe you didn't see before. And so what we've seen, at least that's a good example, is that we took some tandem mass tags. Tandem mass tags are good for isotopically labeling peptides and things, and you can basically determine if something is there, but also quantitate it. And these tandem mass tags, or at least these iota tandem mass tags that I'm showing here, have some characteristic features to them. One of them being that if you perform fragmentation on them, at least with traditional collision-induced dissociation, you get this reporter ion, which is that ring-like structure with the nitrogen in it. And these TMTs have different isotope labels to them. So for example, the one in red doesn't have any isotopic label. Well, it will naturally, but you don't induce it in self. And so that will give you just a monoisotopic peak in your spectrum. What you can do is you can introduce I think this is an isotopic label on the nitrogen here, then you end up seeing an enrichment effect in this structure, and then you can say, okay, if this TMT was binding to this peptide, it must have come from here. And you can do this for a whole bunch of different tags, but what we're showing here is that while these tags are all shifting by one mass unit, previously it's kind of unthinkable to think, ion mobility, can you even get down to separation of single mass to charge units? That's what a mass spectrometer is for, right? Most mass spectrometers can do that, at least the commercial ones, certainly even quadrupoles can do that. But what I'm showing on the right-hand side here is that with our SLIM going up to, I think this was about a 1.5 kilometer separation, you can actually start seeing the differences between single masses. Not all of them, so you can see for, mass to charge 126 and 127, those are pretty much on top of each other, and those are the red and the green colored TMTs. But then there's a shift if you start to look at the blue and the orange ones, for example, and you can see it, and that's quite reproducible as well. And then from there, you can see another shift going from the TMTs that are shown in purple and yellow. And so these are just kind of illustrating how far can you get an ion mobility separation, and if there's a unique feature that you're trying to identify in your sample, we have a pretty good shot of seeing it, at least with the SLIM technology here. There's another interesting piece of work that came out of Switzerland, which uses SLIM coupled to another measurement, which may be useful in helping probe this heterogeneity in everything really, but in soil for sure. And that's how they coupled SLIM to cryogenic IR measurements. So they're actually doing cryo-IR on mobility selected ions. And again, because you can get such good separation between lots of very structurally similar ions with the SLIM technology, well, then you can mobility isolate those and then do IR measurements and see, okay, get some functional group information, and then does this help determine what your structure is? And so on the top here in the pinkish purple color, you see there's a drift tube measurement. There's just one kind of big fat peak, and you get an IR spectrum, and it's comprised of lots of different things. If you then mobility separate these things with SLIM, you end up getting two isomers whose structure, at least with the IR spectra, looks similar to the drift tube. But then if you mobility separate, say the first peak in red, that looks different than the one, the mobility selected ion that it comes later, but it's shown in blue. And so once you see these things, okay, well that gives you some information about your ions. And so one of the things that I wanted to show here is that, well, okay, so we have some ultra high resolution measurements that we think could help. And so on the left-hand side, this kind of just shows the difference between drift tube and SLIM. And you can see the SLIM peaks are quite narrow. We know that people like Orbitraps, though, for mass spectrometry, they're very good, they're very high resolution, and they give fragmentation information. And so the goal of what I was tasked to do here was to try to combine some of the measurements of SLIM and Orbitrap together and see, okay, can all of these gas phase measurements combined help determine structures? And this was just recently published last week, so I won't go into details about exactly what else we can do. But just trying to show some data, does this look interesting to people? So I ended up running some fatty acids, very similar to what I showed on the previous slide. And this shows cis and trans isomers. These are baseline resolved with the SLIM. So the one on the left would be a cis-2 hexadecanoic acid, so a 16 carbon chain fatty acid with the double bond at position 2. You can see that the cis bond is coming out early, which introduces quite a large kink in the chain, and so that presumably arrives early, and then the trans arrives later. You can see that very clearly here. This is fairly routine to get with the SLIM. But again, this was also acquired with the Orbitrap operating at its highest mass resolving power, which is great. And then the spectrum on the right shows what happens if you move the double bond to, say, position 9. Well, the peaks get a little bit closer, but they're still nearly baseline resolved. And so this shows, okay, yeah, so this can maybe help resolve some things that, I don't know, maybe a GC would struggle with this, or maybe an LC might struggle. Something else that was of interest was to try to look at triacylglycerols, again, more lipids. These are, I ended up running just 3 standards. One, can we see it with the SLIM? And then can we separate them? They are fairly different in mass here. So these were about 800 to 880 and then 990 or so. And so those should have been quite easy to detect with just the mass spectrometer alone. But we can see the ammoniated adducts for some of these. And then on top of that, you can also perform a mass isolation and subsequent fragmentation on these peaks to end up getting MS information. And the benefit of all of this is that you can plug this also into, say, if you have a feature extraction or data processing tool that you like to use, well, this should work with that as well. And so we're just trying to make it as easy as possible, though I guess you would have to test that to see if it is in fact easy, because it might not be. And then the last thing I wanted to show you was that this is very new data. This was acquired about 3 weeks ago from a lipid extract from a fungus sample, or at least a fungus isolated from soil, and then was grown in culture afterwards. And they were trying to see, okay, can we see triacylglycerols in that sample, or really do we see anything at all? And there were quite a few features that I'm just highlighting here. But what I'd like to point your eyes to is that we saw a lot of phosphatidylcholines. These are actually pre-charged species, and so they light up mass spectra. And what you can see, at least from the SLIM separation, is that if you see in blue, we actually saw 2 peaks from mass to charge, 786, and then on the right-hand side shows the mass spectrum. But what we ended up seeing is that they have the same exact mass, but there's 2 peaks. And it's like, so what are they? Well, we can do a mobility-selected mass isolation, do fragmentation on those, and then see, okay, we see the characteristic phosphocholine peak, 184, it should be a phosphocholine. And then do we see any of the side chains? In this particular experiment, we'd probably have to turn down the fragmentation energy so we can see some of the chains and attribute, is there an 18 chain or a 17 chain or something like that. But we can see them, and then we know there's 2. And because there's an Orbitrap in there, we also know that these were protonated instead of being sodiated. And so that helps us determine chemical structure with a very high degree of confidence. And then from there, we can start assigning, okay, is this a 36 carbon chain with 2 unsaturations? And then what are the chain lengths in particular? Not necessarily the doubled bond position, we're still working on that, but it is something that we're actively pursuing, and hopefully something like this might prove useful for the characterization of soil. And then the last slide, because I know I'm running over now, I just wanted to say, the process for taking this data, it would be very useful to not have to run individual standards for everything, especially in a complex mixture like soil. And so can we take all this data and run it through a theoretical workflow? And so can we do a theoretical prediction of a structure based on the collisional cross-section, based on the exact mass, and based on the MS. And can we generate a whole bunch of structures from there? We'll probably get thousands. As a matter of fact, we've generated tens of thousands for certain structures. Can we then narrow that candidate list down and filter it based on something? Can it be based on IR? Could it be based on collisional cross-section? To tailor that down, and then from there, can we actually guess based on density functional theory or some other process, what are these structures that we're looking for? So hopefully that is a pathway that people might find useful. Maybe these gas phase measurements could be something that people try to explore, or maybe they're worthless. Maybe somebody doesn't want to use these. That's all interesting information to know, but it's something that we're actively trying to pursue, at least for looking at all these diversity sets of compounds in soil. So thank you.
Thank you, Adam. Now we'd like to transition to the discussion portion of the workshop. We'd like to hear from you all. We don't know how many of you are actively pursuing metabolomics in soil or soil research in general, or even climate-related research. But we're going to put the question slide up that we had earlier, and I'm going to hand this microphone to my co-organizer, Kirsten, and I will take the other microphone and patrol the audience.
Great. So I guess we'll start by opening if you had any questions for our panelists based on their talks that people wanted to address. And in the meantime, you can be looking at some of the discussion questions, and we can kick it off with that if there aren't any specific questions for the panelists based on their talks.
Could we maybe take a poll and just among the audience, how many of you are conducting research similar to, and I don't mean just metabolomics, I mean soil-related research? Okay, awesome. Maybe we could just, any questions? Otherwise, we can start with questions on the screen.
Right, so let's kick it off with the first one of how the chemistry of extracellular metabolites influences the potential for carbon to be sequestered onto charged surfaces of soil minerals. And maybe, Joëlle, if you're comfortable, you could kick it off since that's a lot of your area.
Sure. So I guess I don't have an answer to that, sorry. But I guess from like studies, what we have seen so far is that a lot of the sequestration, or okay, sorry, let me start again. I showed on my last slide this map with like microbial respiration and carbon sequestration, right? So carbon sequestration seems to be prevalent where the microbes are not that active in soil. And soil or microbial activity probably has mostly to do with first external factors, like for example temperature, but then also on, for example, carbon to nitrogen ratio, right? So I think the extracellular metabolites in that respect play a major role, like in how active the microbes can be, and if they turn them over or not, and if they turn them over, if they are respired, or if they are maybe fixed in more polymeric structures that can then be bound to minerals, for example. So I think it's definitely something we need to understand better, especially also in the aspect that we are thinking mostly about soluble molecules because that's what we see relatively easily in mass spec, but especially bacteria, they make polymeric structures as well, like EPS, right? Like biofilms, or plants make these polymeric structures too, which is probably a more stable form of carbon and not that easy to break down. So maybe we should try to take that into account as well.
Maybe a follow-on question to that for Myrna would be to think about, I mean, you've thought a lot about these mineral interfaces, and so thinking about what maybe classes of compounds or how the charge of the compounds might interact with that mineral surface. And then I think, we spend a lot of time thinking about mineral-associated organic matter, but we know there's also like these organo-organo interactions that are probably quite important and prevalent. So I wonder if maybe you want to comment on that.
Yeah, sure. So I think just trying to understand the chemistry of organic matter and predict based on that chemistry the reactivity with respect to mineral surfaces is really challenging. Excuse me. It's dependent on the mineralogy, the ionic strength of the soil, the types of cations are in the soil, the pH of the soil. And there is a lot of evidence, especially using a relatively new technique called comprehensive multi-phase NMR, that specific components like more aromatic type structures are actually more closely associated with the mineral surface, and then you have more polar compounds absorbing on top of those, so you have this layering process. And so that's important with respect to the reactivity, because a lot of what we see using these more like overview type approaches like solid state carbon-13 NMR, which shows you everything, a lot of the aromaticity that you see there is actually not biogeochemically active because it's bound tightly to the mineral surface. So these are kind of the concepts that are emerging from some of these more advanced NMR methods. So that's part of the challenge, I think, when we're trying to determine what we're trying to isolate, because the concept of an exhaustive extraction just isn't going to work with soil. So we need to think outside of the box in terms of what recoveries and things like that. And I mean, if your experiment is constrained, even if it's a field experiment, having a control is super important, because that becomes your reference point. Because getting compounds out exhaustively is probably not a good strategy. Yeah, so I don't know if that answered the question.
I have a question for you, actually. Yeah? Okay. So I have a question to like the mineral person here in the room, because I don't know much about minerals, but in one of our studies what we did is we took exudates and we had these like plants that were growing on clay granules, and then we found that most of the exudates were actually bound to the surface of the minerals afterwards, and we couldn't detect them as soluble compounds anymore. So I wonder, obviously when you like have clean clay and you incubate plants, then they have the capacity to bind a lot of exudates. But I wonder how it is in a natural soil system, if you have a feeling for how saturated or not so saturated the minerals are, if plants make exudates, if most of them are probably then immediately bound to clay if there is there, or if clay in soils is saturated anyway, and if there is like a chance to use these surfaces for carbon sequestration at all or not.
That's a great question. And there's this concept of carbon saturation, right, for ecosystems, and whether or not are our ecosystems currently carbon saturated, or can we put in more carbon? And I think what I'll draw on are the litter manipulation experiments that have been going on for many decades, where they have taken double plant litter, so above ground litter, and added it to the soil. And over 20, 30 years, there's been no net increase in carbon. But it's more likely due to priming, which you talked about, rather than, these new compounds being able to form that part of the mineral organic matter, complex. So I really don't think we have a very good handle on how dynamic the mineral organic matter complex is, but we do know that there are layers of organic matter compounds sitting on those clays, and it's variable depending on the properties of the soil. We also did, using comprehensive multi-phase NMR again, we looked at biomass clay interactions, and we could see that bacteria and fungi could attach themselves to clean mineral surfaces, like whole cell attachments. So I think that is interesting in the sense that, do microbes have accessibility to these compounds when they're part of that complex? They possibly can, but clearly we need more work.
Could I ask a question to follow up on that? And this might, I'm a metabolomics person, firstly, not a soil person, so this might be naive, but my thinking is that if we were to be able to successfully exploit soil as a sink for atmospheric carbon, then we should know, ideally, all of the chemistry that's present in soil in order to then potentially build a model or to be able to predict which of that chemistry is going to bind irreversibly or become otherwise inaccessible via interaction with the minerals. And so then my question is, maybe it's not a question, maybe it's a speculative statement that we probably know very, very little. I mean, we know a lot, right? We know certain chemicals that have been structurally identified, and we know how they might interact because we can spike those in. But, you know, if you just went out, if we walked outside here and took a sample, how much of the chemistry would be known there in terms of its interaction with minerals that would be present in that soil?
That's another good question. I think it depends on the technique you use. Again, I'll go back to NMR strategies, which will provide a good overview of everything, but then when you're dealing with a complex mixture, you have overlapping resonances, so it is challenging to say that, you know, this layer is X and that layer is Y. And then the more complementary methods are using different extractions and then using GCMS, LCMS, et cetera, et cetera. So I think using those hand in hand is also important, but then to get, the nice thing about NMR is you can literally just put the sample in the NMR as is, but you have that complexity and the overlapping resonances, but then the other strategies require extractions. So how much are we actually getting out and into the mass spec, like after separation and ionization? I think that's a problem. So I think NMR gives a good overview, but not the resolution that we need, I think, to move forward. So yeah, but we can still see pretty much everything from a general sense, but not the fine chemistry that we need. Yeah.
Not the chemistry, not the detail that would enable us to make predictive models on how to force it through into inaccessible forms.
Yeah.
I've got a question here in the audience. Arpana.
Thanks, Tom. Great workshop. It was really informative. I don't do soil, so I learned a lot. I wanted to switch gears here to talk about like the reference-free compound ID strategies that Tom had mentioned in the introduction. How do you propose to get people on board to do such, you know, workflows or strategies, especially when there's like a huge conflict of people who only trust data that's backed up by standards versus, you know, this new paradigm that was introduced?
Well, that's a problem, trying to get people to do that. I can tell you, even just trying to get people to use a Slim. You go, you want to use a Slim? They go, no. And you go, all right, that answers that question. So I think that you really have to back it up with some concrete evidence, and there has to be a lot of it too. This kind of goes to the question number 4 here, which I also think was directed at me, is the reference-free paradigm ready for prime time? And the answer is, well, we're going to need to get a lot of data to show. I didn't even show any examples of a prediction yet. And I think that's because we're still trying to figure out, well, what is the best, is there a best workflow, one? And then if there's not a best workflow, what is maybe the optimum one for different sample sets? So do you have a better one for lipids than you do for metabolites, than you do for peptides, that type of thing? But you do have to have a lot of good demonstrations, and you know, initially, yeah, we're gonna have to back them up with standards as well. That is what people trust, and I think maybe slowly but surely stepping away from that. You know, maybe you do a mixture and you say, you know, there's 20 things in here, and you verify it with 19. And you go, well, what is this one left? And you go, well, here's how much information we can get. We can get some ion mobility data, some mass spec that people can trust, but then also, you know, maybe some IR, and then put these into some computational tools, which people are also going to have to trust, which is hard. And once those get into probably readily accessible forms where people can go and say, press a button, and you can do that on your desktop or your laptop, however you want to, and then just do it enough times to convince yourself, and it doesn't crash, it doesn't throw an error, it doesn't give you a website that you need to go to for help. Maybe that can be a little bit better. Some of the things I run into with my instrument is like, oh, hey, I can give you as much data as you want. And they're like, that's great, but I can't use my processing tool for it. Oh, Great. So then we have to go and figure out how to make it amenable for somebody else's processing tool. So it's really a whole development thing. But yeah, I think it's just you're going to have to convince with overwhelming evidence, and then not only that, but do it on something that people care about. So, can it be, lipids are great too. I'm certainly interested in lipids because they're also an analytical challenge, but, what would be also relevant to society today? Are there things like medicines? Do people care about medicines? I don't know if we can do an enantiomeric separation with ion mobility ever, unless you do a complexation reaction, but there are enantiomers where people would be pretty interested that, one enantiomer is a therapeutic one and the other one kills you. So we don't want to, have any of that in there. And so if we can at least, make something and say, all right, so there's some evidence that one is present, people go like, oh, okay, don't use that one. So maybe that.
It's completely unrelated, but maybe it's slightly related. And that is, we've had this idea that you should be able to predict structures out of gene clusters and synthesize them. And there was significant pushback against that. I mean, for 5 years, we didn't get a single grant funded. First papers went out, and we were told that'll never happen again. You'll never be able to get that to work again. So I think it's just a matter, if you have a new paradigm, you have to have a good discovery. Like when we finally found a molecule that had a new target, right, that someone actually cared about, then another person would do it, another person would do it. So anyway, it's unrelated, but it's the idea of you're using informatics to do something that one thinks you should be able to do with reference. You should have a compound there that you can look at and tell us what it is. It took us a long time for anyone to believe it would work, and it really took a single paper that had some interest to people where people would then say, they still don't believe it. I will say we still have good papers that go out, they get half the reviewers coming back saying, this is bullshit, this is BS, there's no way it's going to work. So, but I think you just have to have a good discovery, right? You have one or two good things that people outside of your group care about, then it begins to pick up some steam.
Yeah, I think for me, as mostly a user of metabolomics, that's really the key point, right? That with the untargeted methods and the bioinformatics, it's kind of a hypothesis generator, right? That you try to figure out what are the, I don't know how many % of things that we don't know that would open up new directions and novel ideas, and then you kind of postulate what you see, or you describe what you see and your best guess of what it might be. And then in a second step, you verify, right? But it's usually a two-step process because it's a lot of work generating that, and then you have kind of an idea or an overview, and then in the next step, you need additional grants or whatever to actually look into it, right? So I think people need to allow for this, of the more computational way of looking at that first. I personally find as long as you clearly communicate what you actually do or how you did, that's totally fine, right?
Question from the audience, Vasu?
Thanks, Tom. It's not a question, it's just a follow-up on the discussion points we just had. So I think the field of metabolomics needs to follow somewhat what, protein structure biology, especially the computational side of things, they follow, right? Because they started with simple protein prediction, structure prediction, and then went on to, now we have AlphaFold 2, yeah. So everyone is trying to hop onto that. So I think metabolomics as a field also just need to follow the same stream. What Aparna was also just mentioning that, yeah, where does it come? Like, maybe we need to have the standards comparison, yes, but then we need to start from somewhere. And also another thing, like we just saw great talks here from Metabolomics and then spectral prediction as well. I think we maybe just not need a website or two. We need an integrated platform or tool where all the studies can be integrated together, and then probably an end user can use it, and then maybe it'll just start to have, that belief system into the user that yes, yeah, data integration is there, data enrichment is there, then we have prediction sets, maybe the comparison as well. So as a whole, we do need a platform, like a centralized system where, we can have all the data which is being generated or will be generated, and then to have all that studies done. So yeah, that's just a point, yeah.
Thanks. I'd like to follow up on the question of the reference-free and the question of the statement of moving towards what has been done with other omics. And this might be a little bit of a random rambling statement, but I'll try and bring it home at the end. So, battery? Okay, I'm back. I conduct proteomics research, or I apply proteomics in research, as well as metabolomics. And so what is done with proteomics? We typically will take we'll use mass spectrometry. We'll use tandem mass spectrometry to sequence peptides based on how those peptides dissociate in the instrument, and then we match the experimental data to essentially predicted data using any number of tools. But how do we know which proteins and peptides to look for? We refer first back to the genome of the corresponding organism. And so if I'm doing a human proteomics study, I will rely on resources that have been developed by the proteomics community, which primarily, number one, is a reference protein file that's based on the genome of the human. And we do something similar for metabolomics. We do use reference libraries, but these are reference libraries that we all build ourselves based on whatever we can get our hands on in terms of molecules. And so if I use an analogy for soil, then how do I build an appropriate reference library for soil? Well, first I start with probably my human metabolome library that I've built for human studies, which wouldn't be appropriate for soil. I mean, there will be some primary metabolites that are common between the two, between humans and what might be present in soil. But I think my question then comes back to what Sean presented on, and could we build appropriate reference libraries for any metabolome based on biosynthetic gene cluster analysis in scale, and then using those analyses to tell us which molecules we should look for or put into our reference library?
Yes. No. Okay. So I think there's actually two problems, right? It should be divided into two. When you think about at least biosynthesis, it should be primary metabolism and secondary metabolism, right? And I think that the thing that's probably going to dominate in soil is primary metabolism. It is very similar to what the human microbiome people are finding. There was always this postulation the human microbiome would have all these biosynthetic gene clusters in it, and that's what you should look at. Turns out they don't. All these things I told you about don't exist in the human microbiome. It's really just shunt products of bacteria doing metabolism that are the bulk material you're seeing in there. I think we should leverage that information to first go out and sort. There's a huge community that's spending a lot of time characterizing those to make libraries of what the gut looks like. My guess is that's going to be a first layer approximation of a little bit of what the soil looks like. And so I think there's something we could do there with primary metabolism that would be very informative and largely interesting for soil. Because secondary metabolism, I don't know, we can predict things, but there's going to be very little of it in there at the end of the day. And so I would divide the problem into two parts. And I would, if I care about bulk what's going on in soil, I would look at primary metabolism to start with and focus on our predictions there, generate references of all the things you think might go there. I would steal from the human microbiome because there's a lot more money there, and there's a lot of people doing work there, and the primary metabolism is probably going to be pretty similar, and then add whatever's left over. And then I would tackle the secondary metabolism afterwards. I don't know if that answers the question, but I think the primary metabolism we can do. And it's probably not that complex, and it'll be a great place to start. It's going to be the bulk stuff.
Yeah. You know, I may have previously in years been, I wouldn't say misled, but also under the impression that the secondary metabolism should be where the focus is. But it's interesting to hear this perspective in context of what's been done with the human microbiome, that primary metabolism perhaps should be where we start and where we focus on.
I think so. I mean, again, if you're looking at communications between organisms, then I think you're probably looking at secondary metabolism. If you're looking at bulk, what's happening, the carbon's being turned over, and how the bulk product, how sugars are in there, how acetate's being turned over, what's going on with bulk amino acids, that's all primary metabolism. And I think so. If we're looking at the bulk kind of carbon sink, if we're trying to ask how things communicate and talk to each other, then it's secondary metabolism, I think, is going to come a significant role in there. But if it's just what's the major kind of metabolite storage transfer, I think we're going to focus on primary metabolism, and that's a doable problem. Not everything, right? There's going to be bugs doing strange things, but there's a whole lot to start with where your set could be pretty well defined, and you'd have lots of things that we actually have hard copies of. We have real molecules, and other things you're pretty sure you got the prediction right, and it would be a reasonable place to start. And most of that major stuff, I'm going to guess, is in that set, and it's going to be people really caring about how the root interacts with the microbe and how a microbe interacts with that are going to switch to secondary metabolism.
Interesting. Thank you. Kirsten.
Yeah, I have a follow-up for that. That primary metabolism is happening within the cell. And so it's curious, or if we're seeing exchange between the plant and the microbe, right? And so it's curious to think about how, from a metabolomics approach, it's hard to like capture that metabolite exchange or even what's within a cell because of the challenges that Myrna talked about with like extractions and getting things out of the soil. And so the genomes become interesting because you may be able to decipher that easier than with actually capturing the metabolites themselves, especially if they're quickly exchanged. And so I guess I want to follow up on how this genomic approach could work, especially considering one of the big challenges is that we want to look at different phenotypes, right? So just because we have that genomic potential there doesn't mean it's necessarily being expressed under different environmental conditions. And if we want to change the carbon pump, right, then we actually have to understand that transcription part of it.
Yes, it's a great question, and it goes to the heart of the problem. It's all these things interacting, right? But I think at a basics, genomics is the easiest part, right? Just sequence it, and that's where we are today. And where we go from there, probably transcriptomics is the next thing to go to, and what's happening in the soil. And then the next thing is probably metabolomics, right? I would guess, just because the first two can just be done on one machine. You just put them on there, and you just look at them, and it just becomes an informatics problem of trying to interpret that data. The metabolomics, it's how do I get it out? Everyone's going to look different. I mean, when you look at DNA and RNA, it all gets treated the exact same way, right? So it's just one experiment, and it becomes an informatics problem downstream. Hit metabolomics. How do I deal with lipids? How do I deal with charge thing? How do I get them out? How do I do this? How do I do that? And so, yeah, it would be great if we could get models of the first two that helped us with figuring out how we should be tackling the next one. But yeah, I think I personally think we're a few years away from figuring most of it out. I think the sequencing is in the near future. I think how one does all the metabolites out there is challenging, and then of course how you predict the expression is we don't have any clue. I would argue we don't have any clue of really how we're going to predict which genes are turned on and off. It's just going to be sequence it and figure out, oh, these are on, those are off at the moment. I don't know if I answered your question, but because I think the answer is it's complicated.
We have a question here, but I just want to make one statement before I pass the mic over. Sequencing is awesome, but everything we do with the sequencing today was the result of about 12-year directed project with a total budget of about $3 billion in the form of the Human Genome Project. So maybe it's time for the Human Metabolome or whatever, Soil Metabolome Project.
Hello. Thank you very much. Very interesting talk. I have a talk too on Thursday about soil metabolomics, and I hope you can attend. So I have first a question and then some considerations. The question is, I was trying to finding a tool to annotate non-ribosomical peptides, and as far they have some weird amino acids, some tool doesn't work. So do you have any solution for that?
Maybe you could borrow some techniques from the proteomics community in terms of de novo peptide sequencing? Okay. Yeah. Mm-hmm. Okay. Yeah. So some people that I can think of would be Pavel Pevsner at UC San Diego. He has developed for a long time various computational tools for interrogating proteomics data, and a lot of them do lean towards the de novo, meaning you determine the peptide sequence directly from the data and not in comparison to any reference. Yeah, so I would check for some of his papers, but maybe just doing literature search in general for, okay, yeah. Another person to ask would be Sam Payne. He was a former P&L colleague, and he's now at BYU, Brigham Young University in Provo, Utah. He has done a lot of proteogenomics work and tool development, and he might not have a tool or a solution, but I know he'll have an opinion. Yeah.
Are these proteins or these small molecules you're looking at?
Yeah, are small peptides, so like 10 amino acid or 15, etc.
So
like
there's the
tables.
if you want to know every ribosomal peptide building block that's ever been reported, there's the Norine database.
Yeah,
So
exactly. the Nordic
that'll have
database,
everything
yes.
that's ever been seen.
Yeah.
There are going to be things that haven't been seen, but if everyone's ever been seen,
Yeah, but
relatively well curated.
when you do soil metabolomics, you find a lot of these things that doesn't have a match in Nordic database. So it's kind of, if we want to discover some more, that will be a good tool to develop. The other thing is, okay, we do metabolomics in soil and we apply a completely reference-free compound identification. So we do everything in silico we try to do MSMS approach for what is known, and then we apply in silico prediction of the mass spectra, retention time prediction, CCS prediction, actually the one from PNNL. And then we try to annotate the maximum molecule as we can, and we reach 20–30% of the MSMS we get on the sample. And actually, we participate in CASMI past year in Valencia that are pan-organized, and we get right 23% of the identification. And that was the best one, because the best one was 25%. And was about plant compounds, and plant compounds are strange ones. So it was like microbial metabolites. So I think it's very comparable what we get. So we're talking about 25% maximum of correct identification in soil metabolomics too. Is that enough? From our experience, I can say yes, because you get something that is actionable. So we do analysis for farmers, and actually we want to give some information that actually they can action on that information. So for example, pathogen that are in the soil. So pathogen produce a lot of secondary metabolites. I don't care about primary metabolites on the soil, I just care about secondary metabolites that I can link to specific microbials. So that's my challenge. I can identify specific metabolites and then link to this. So I am a fan of CCS, I have an ion mobility system, but it's very challenging. So it's kind of community effort that should be on there. It helps a lot that there is now this new library like Lotus. I don't know if someone here is present, is curator of Lotus or MP Atlas, that are great because you can link the taxonomy to a specific metabolite. And also you can use that library to actually find in your soil sample. Probably we get a lot of annotations that are wrong. I'm sure about that, 75%. But the 25 that we get right, I can see also the advantage of metabolomics. I will talk also that the advantage of metabolomics from metagenomics approach. Because in the same analysis, you can get plant compounds, you can have information about the interaction. That is very important. So that's it.
That's a lot. Thank you. I have a response. AV guy, sorry, I didn't get your name at the beginning. Could you put up the slide with the QR code? So while we continue the discussion for the last 10 minutes, this is a link to a poll. If you would be interested in participating in the position paper, then please scan it and indicate your interest and provide your email, and then we will follow up. One comment I wanted to say about CASMI contests, awesome, but to date, they've only included MS spectra. Is that true? Yeah. One? In that one. So that's a big advance over previous CASMI contests, which had only, up until Valencia, included tandem mass spectra. Mm-hmm. Yeah, yeah.
but not highly used. They were, optional, but no CCS data or anything else. No NMR. Yeah, and one of the things or challenges that hasn't been addressed in a CASME contest is doing a real unknown. Like, how do you even start with that? Because most of the times it's probably going to be structural dereplication. So that's a little bit challenging for the contest itself, but would be a great addition to really test, have a real test in that case.
So I'll come over here. I'll talk while I walk. So one of the things that I've thought about in how we conduct a metabolomics experiment, I think metabolomics, modern technology and methods are inspired by proteomics. We've seen what LC-MS can get us in terms of protein identification, and we try and fit that to the metabolomics challenge. But I believe that metabolomics is a round hole and LC-MS is a square peg, and we need more than just that. And so I'm personally a huge fan of multidimensional, as many dimensions as we can get in a single analysis.
Hi, this is just a kind of comment that I just want to throw out there to the speakers as well as the general audience. I'm fascinated by the reference-free structural elucidation as a topic, but I'm actually not a metabolomics expert. I'm a mathematician, actually. I work on machine learning and deep learning models, and I've been building just various mathematical models on reference-free structural elucidation on my own, and I would love to collaborate with anybody here who's interested in implementing deep learning models in this field. I need a lot of guidance, of course, from the life science folks. So I would love to chat with anybody here who's interested in exploring that topic together. Yeah, my name is Richard Kim. I'm working for a company called Dice. It's a food company.
So I don't know if you know, but in the literature already, there are a number of tools. Yeah, you've probably seen it. Yeah, okay, cool. So the current tools for what we would say reference-free metabolomics are all what we refer to at P&L as the forward direction structure to observable. What would be the holy grail would be to go from observable confidently back to structure. So go from your ensemble of data back to a chemical structure. Did you have a question? No? Yeah? Okay.
I have some technical questions. So when you use the SLIM, you said that the path length is 1.5 kilometer. So in that case, if ion move like that long path, then some of them exhausted and they don't want to go inside of detector. So do you think that that might affect the limit of detection of certain type of molecule, or?
Yeah, I think the question is, the technical question for the SLIM is that if your ions are going around a very long path, do they ever dip below your detection limit? And the answer is they can. There are some ways that we have to get around that. We can play with some of the traveling wave conditions to try to make this diffusionally broad peak go back into a single peak. You can do that by changing traveling wave amplitude. Like I said, we have traps. We can do it like an ion trap. Traps are good for accumulating lots of ions and then throwing them out all at once. So that can help as well. I actually wanted to address that machine learning question as well. This is one of the up-and-coming technologies that we are certainly exploring. It's pretty powerful. And one of the things that we have to consider, especially when we can start to probe these complex mixtures with more and more detail, is that if you have these techniques which are used to looking at a single peak, how do they perform when that peak is no longer one peak? What happens when it's split into two now? And then if you've trained your model on one peak, now you're gonna have to go back and train it on ones that have two. And then if you do that a lot, is it only gonna look for two? And so there's this kind of recursive effect. So, I kind of agree with the fact that, yeah, it's gonna be a bit of time before we can really get a good workflow in so that we can convince people that it's working. But these are just the types of little things that keep popping up that we have to keep addressing. And maybe there are 25% of identifications it's good enough for some people, and then the next day somebody walks in and goes, that's not good enough for me. And because now I see 2 peaks where we're only seeing 1. And yeah, it's just going to be this recursive thing until we can get enough that most of the community can be convinced. And then of course you're going to get somebody who comes in out of the blue and says, well, it's not good enough for me. So then we have to keep going and kind of roll with the flow there.
Yes. Hi. Thanks for all your nice talks and very nice discussion. But to comment on that, that is assuming we actually see peaks. We can extract all the relevant molecules from the soil. I noticed one of the questions was, are there any innovative ways of extracting the molecules? Because I know for genetic material, there's a lot of development, and I think as someone showed, we can see more and more, and we start to wonder what that is, or sorry, what the genes are producing. But for molecules, I've been browsing the literature, thinking about proposals, how we can, find more of the dark matter in the soil. But I haven't seen a lot, so maybe someone in the audience or up there, do you know more? Can we actually extract most of it now?
Part of the challenge, though, is that some of it's non-extractable because of the association with minerals. So, I mean, if you demineralize the sample, you can get out probably between 70–80% of all of the carbon. But they're, using something like hydrofluoric acid is really contentious, even though it's really all we have right now. So there are like a number of methods out there. So for example, there are different hydrolysis types of methods, base hydrolysis, acid hydrolysis. But the issue is that some of these techniques will yield things like amino acids, and that's not necessarily informative if you're trying to trace processes or assign a source. So that's a challenge. I think there are probably some methods out there, but again, I mean, should we really be changing the sample to fit the analytical technique when we could possibly push the analytical window more and fit the analytical method to be more amenable to soil samples? I personally think that's a better pursuit. And again, I'm not, comprehensive NMR is why it was developed, because we were tired of changing, the probe 3 times to isolate different components of the soil. So by developing a new NMR probe, we can now detect everything. So I think personally that's where we need to go, and I think we need to push the analytical vendors to be more holistic in their methods. But anyways.
Yeah, I agree. I think another interesting opportunity is potentially pyrolysis GCMS, and then if we can use, and I'd love to hear your perspective on that, Myrna, but if we can, use combustion at different temperatures and ramp that up to look at the strength of molecules. Again, we have the problem of we can combust and we can get a bajillion peaks, but annotated. So, but that's another method that's it's destructive, but you don't necessarily have extraction bias, and so we have a little bit more control over that.
There was another question up front.
Thanks very much, everybody, for the workshop. I found it fascinating, and you've given me a lot of nightmares about extractable metabolites and things. So I think you're beginning to answer some of my question, but I did get the impression that maybe we should be thinking about other technologies that were designed specifically for surface analysis. So I just wondered whether anybody is doing MALDI or DESI at the moment off soils, or whether, does the IR or Raman spectroscopy tell us a different story to what we might be seeing in the LCMS-based approaches? And then one nightmare, just to give back to everybody, is are we also seeing non-enzymatic reactions that are happening on surfaces as well? So things that we can't predict from gene clusters necessarily.
So I can add one comment for exudation. and we started to play a bit with Maldi. The issue though is at least plants get really big really fast, so we are kind of limited in how much area we can screen over time or how we can pick what to look at. I know other people started to look at roots in soil as well, and they definitely see patterns. Again, usually the issue then is with annotation because we don't get the same, or we don't get fragmentation spectra usually. But there are definitely approaches or people looking into that, and there is definitely a lot of spatial dynamics going on. So I think it's very valuable and additional layer that we have to take into account. I don't know if somebody else has comments on soil.
Yeah, I think that you know, the surface imaging techniques are good. I think with DESI in particular, you're still kind of limited to, I think you're even more limited than what you can do with just a regular extraction paste. You're really limited to what you can electrospray, which would be water, methanol, acetonitrile, kind of those things. And unless you have a different way to work with a different extractable solvent. But I think in that aspect, you know, if something's not going to come out of your soil sample with those solvents, kind of no point to use DESI, in my opinion. I mean, you'll get the surface coverage if you're looking for spatial resolution, but yeah, I mean, if something comes off only with hydrofluoric acid, I mean, that's not a good one to electrospray. Same thing with like hexane and stuff like that. It's more difficult to work with. You're probably just better to throw the sample in a vial and extract it, would be my opinion.
So technically, we're at time. I mean, it's been great discussion so far. This was a 2-hour workshop. It's 2 p.m. Eastern. I don't know how everybody feels. I know some of us would probably be willing to keep going depending on degree of discussion, but you can stay if there are more questions. We don't want to hold you. I think we leave it up to the audience to decide whether they've had enough or not. I don't see any other questions, so yeah. Thanks everyone for attending, and thank you for participating. It was an awesome discussion.